has it correct. Nazila said, if we look at this image, you can see that actin is all over the place, but that doesn't really give you a clue because the red is kind of all over the place. But cortical actin, what is cortical actin? Around the edges. So the fact that you can see the edges of the cell with these green fibers tells you that green is actin.
Okay.
Okay, does that make sense?
Yeah.
And the red, what's red then?
Intermediate.
It's actually microtubules,
Really?
and it's hard to tell that, right? What did we say microtubules should look like?
Hollow.
Okay, so if I draw a cell, and here's the nucleus, right, drawn incorrectly, I look at, wait, who was I just saying? So I need a device too. So if I were to draw microtubules, they should look like, what was the thing I said they look like? Spokes on a wheel. Okay, spokes on a wheel. Emanating from where? The MTOC, otherwise known as microtubule organizing center or centrosome. The centrosome has what in it? Two centrioles. Okay, so now take a look at the red in here and notice that this is the simplistic version, yes?
Mm-hmm.
It's a little more complicated. What's blue over there?
The nucleus.
Nucleus. So how do we stain for the nucleus? Anybody know what stain we use? No, hematoxylin doesn't stain nucleus.
Does it say DNA? There you go.
I'm right? Mr. Muselli, I'm right? Oh my God, mark it on the calendar. Okay, how do you stain DNA? Who works in a lab? Dude, how do we stain our DNA? What's that red stuff we put it in?
Oh, gel red.
No, we don't use gel red. We like more toxic than that. Ethidium bromide. Who said that? Thank you. Are you the other Nicole? You've moved again. So now we're back in a line. We have, well, it's kind of a zigzag. 1, 2, 3, 4, 5. Thank you. I really have never had 5 Nicoles or 5 of anybody in class. So ethidium bromide. And what does ethidium bromide do? We've talked about this already.
Intercalates or base pair.
So the word is intercalate. Intercalate. Inter is between, and it intercalates between what?
The bases.
The base pairs. What does that do to DNA? Ultimately. It can break it. It can break it, but so is it a good thing for you to stick your hands in ethidium bromide? No. It's a carcinogen, a teratogen, a mutagen. Okay, we love it. It's really good for you. Okay, don't be drinking any of the ethidium bromide in the lab. Okay, so the nucleus is stained with ethidium bromide. What is the green? Probably
Green.
GFP. What does GFP stand for? Green fluorescent protein from jellyfish. Thank you, jellyfish. Jellyfish are good for something. Okay, anybody know what red might be? You know what the red stain is? It's rhodium. Okay? Good. All right. So where we had left off was we were beginning to talk about the different kinds of drugs that can affect different filaments. And we're going to talk about both actin-specific drugs as well as microtubule-specific drugs. But then, what about intermediate filaments? What's different about intermediate filaments from actin and microtubules?
It's non-polar.
Nonpolar, meaning it's
Does it have two different sides?
it doesn't have two different sides. It's not dynamic. It doesn't change very quickly. It's structural. It's there to stay. That doesn't mean it can't be broken down, but it's not polymerized and depolymerized quickly. Okay? So these are 3 different drugs: phalloidin, latrunculin, and cytochalasin D. Right? Those are hard words because they're drugs. Phalloidin, it comes from mushrooms. Okay, this is the reason they say when you're out in nature, don't just pick up anything and eat it. The mushroom has another name. Do you know what the other name is? Fungus. The fungus of fungus.
Psilocybin?
It's called the death cap. Okay? And the reason is because if you eat them, it binds to actin filaments and causes them to become stuck. They can't polymerize or depolymerize. And why is that bad? Stuff doesn't move. What doesn't move in particular that would matter? Where do we say actin is found?
The outside of the cell.
It's inside of the cell,
Yeah.
on the cortical edges, so cells can't move. I'm thinking in a more broader context.
Neutrophils.
Neutrophils and this are the same. I heard it. Muscles. What happens if your muscles, right? So what are muscles made out of? Okay, let's draw a muscle again. Okay, remember how we did this? We draw, what are these? It's a Z-disc. Okay, and there are things here. Okay, does anybody know what those are called? Those are the thick filaments, and what are they made out of? So the thick filaments are actually made out of myosin, which we haven't talked about yet. So let's see. How will I do this? Oh, the red is myosin, and they're called thick filaments. And then what does the green? What's the green? That's the actin. Where is the plus end and where is the minus end if they're attached to these guys? Does anybody have a clue where the plus end is and where the minus end is? Do they have a plus and a minus? Yes. Okay, they always have a plus and a minus end. What does that mean, the plus end? So it polymerizes and depolymerizes more quickly from the plus end than it does from the minus end. What does that mean for the critical concentration? It's lower than the critical concentration of the negative end. Okay, so those of you who are sitting there and are a little bewildered, okay, between now and Thursday, you want to get that down. Okay, that needs to be in your head. Anybody want to come draw that? Come on, Donna, you know what you're talking about. Draw that chart, draw that graph for the CCs. You put a new slide in and draw that for us. So I really want you to understand this concept. We were talking about actin and myosins and where the plus end is, and I'll just cut to the chase. So this is the plus end and the minus ends are at the Z disc. So these are all the plus ends. Okay. And all the minuses are on the Z disc. Did I happen to mention what the Z disc is made out of? So this is the Z disc. It's not made of actin. Because it can't move, it can't change, it's really structurally stable. Guess what it's made out of? Intermediate filaments. That's cool. Did you get it? You're a genius? I heard you. You knew it. Yay! Okay. Okay, Stephanie.
So is it just the outside lines that are actin?
So the inside lines are actin.
Okay.
This is a Z disc, this is an intermediate filament. So the actin is attached to an intermediate filament. What's on the other side, right? If I were to keep drawing that, right? Oh, sorry. You have the next Z disc is over here, right? And what is this structure called? The whole thing, a sarcomere. Okay, let's get back to phalloidin. Phalloidin is a drug that's in the death cap. It's in the mushroom. You eat it. Why do you die?
Because your muscles stop moving.
Because your muscles stop moving.
Your
So
heart.
Your heart.
Your heart is
I
a
am
muscle.
stopped moving.
Are you still moving?
Your heart, I'm not really moving. What's moving?
Your diaphragm.
My lips? My lips never stop
Your heart
moving.
is
Yes.
beating.
My heart,
Your
what's the
diaphragm.
most important thing? Your heart can go for a long time. It's your diaphragm. Your diaphragm is what's allowing you to breathe. Okay, and when that diaphragm stops, you stop. Bye-bye. Okay. Okay, latrunculin is another one of these drugs. Okay, and notice these are slightly different. This binds and stabilizes it, so it causes the actin to stay stuck. This guy binds to subunits and prevents polymerization. So if I put this in a cell, what's the cell going to look like? Is it going to have actin in it? Yes, because it's just going to fix it just like it is. But what happens if I treat a cell with latrunculin? Makes the actin go away. Okay, so actin depolymerizes and it binds to all of the subunits and then it can't polymerize again. Okay, this is in a sea sponge. What's a sea sponge? It's an animal, quote unquote. I don't know what that means and I don't care because I only deal with things that are like 1 or 2 cells. I don't even want to do organs, right? I just want cells, individual cells. So why in God's name does a sea sponge make something that's toxic? It's a defense mechanism, okay? Fish comes along and eats me. Okay, I'm dead. But what am I doing? I'm killing the fish. I'm taking him out with me. Okay, you're going to eat me? Ha ha, you're dead too. I'm saving the rest of my fellow sea sponges. Because the fish says, holy crap, right? They're in a school. And the school says, oh, that guy took a bite out of that sea sponge. That doesn't look, he's dead. Right? So fish know. Rats know, yes? Not that I have a lot of experience with rats, but I do have a lot of experience with rats in my house. In my house, did you hear that? That's wonderful. What do rats do? You can't kill rats. What have they evolved to do? Reproduce, but they eat a little bit of something. They only eat a little bit. They won't take all of it and eat it. They eat a little bit and they see, does it make me sick? So the things that you have to get to kill rats are really toxic. That rat bait stuff, that is really toxic. It's like, it'll kill your pets. Okay, if the pets eat the rat poison, the rat dies, and your dog eats the rat, your dog will die. So if you care about any other creatures, don't use that rat stuff. Get the big old rat trap. Have you seen the rat trap? It's a mouse trap, but it's like this big. I'm afraid to even set it. I'm afraid I'll lose a finger.
You can get pet-friendly rat bait too.
How do you get pet-friendly rat bait, and what does it do?
It doesn't cause the bleeding out because that's what like the rat bait does. Is it changes the clotting factors?
Yeah, it causes them to hemorrhage. It's really lovely.
Yeah, it's horrible. But there's pet safe.
Pet safe.
And
So yeah,
I've seen dogs eat it and come in
And they're okay?
Their clotting factors.
Yeah. I'm all about just trying to keep them out. That's my goal. I'm not going to get rid of the rats. The rats are here to stay. Yeah, they like my avocados. As do the raccoons last week. 2 raccoons in the front yard. The dogs went berserk at 3 o'clock in the morning. Absolutely. And I look out, I'm like, oh, I see one raccoon. Then the other one comes around the tree. I'm like, whoa, they're big. They're scary. Okay. All right, are we good with this? The last one, cytoclasin D. All right, it does similar things. It binds to the monomers. About the filaments. I like what you're doing. Okay, and what's the x-axis?
The x-axis, I believe, is the concentration of the
Of what?
G-actin available.
Subunits. So, right, if we're talking about actin, it's going to be the G-actin. If we're talking about microtubules, what are we talking about?
The alpha beta.
The alpha beta.
Should it be on the vertical?
Nope, the concentration of subunits is here,
And the
because we're going to, right? And so the concentration, right, is what matters as far as where we're going to add as far as the critical concentration, right, Nazil? Okay. So then, what's on the y-axis?
Time?
Well, I'm asking this question, and you know what? I don't remember. Elongation rate? Are you looking it up? Yeah. Look, she has elongation, the rate of elongation. That kind of makes sense. But let's see if we can't figure out why that's correct. Okay. So draw for me the rate for the plus end. Make it, that's good. So it's actually a straight line.
Okay.
Isn't that irritating because you have to erase because we're doing the recording.
Okay.
Make it red. There, that's pretty, okay? And so what is rate? What is rate?
Slope.
Slope. What is slope? The change in y over the change in x. Remember that from
Middle school.
4th grade, 5th grade, 6th? I'm not sure. Was it
I think it's
junior
middle
high?
school.
Maybe? Depends how much of a brilliant person you were, right? So some people have it probably in 2nd grade. Wasn't me. I can't even remember when I had it. Okay, and so where would you put the critical concentration?
Like right here.
Okay, and now what about and make sure you label that the plus end. All right, and then what about the minus end?
This one's more logarithmic,
Do green.
isn't it?
It's not, they're both linear.
They're both linear.
Yeah.
I don't know.
Okay, stop.
I
Now,
stopped.
are you stopping? Look at me. Okay. How different are these rates?
Very.
No,
Not
Their
very.
slope is.
they're not at all. The slope is exactly the same.
Okay.
Okay, so you're doing good, all right? So when we're talking about the slope is less, okay, it's going to be basically, there, you got it.
I'm not
Green,
drawing
green,
yet.
green, green. There you go. Okay, so the slope is less. The change in y over the change in x is lower. Okay, and great. So there is the critical concentration for the minus end. And where does treadmilling occur?
Right here.
Let Donna do it. It's okay. That's good. Treadmilling. Great. Donna, you win the prize for today.
Sweet.
That's it. That's all you get. Sorry. Okay? I wish I had my candy with me. But then it seems like really odd, like candy little girl. That's all bad, right? Pretty sure I'll get fired for that. So between these two concentrations, we're above the critical concentration for the plus end, which means, what are we doing to the plus end? Adding. And we're below the critical concentration for the minus end, so we're removing. Okay, and we draw it like this, and really it might be much closer, okay, but we're drawing it so that you can see the difference. Okay? What might make subunits available or not available? If you're all a bunch of G-actins, what might make you available? Okay, it seems right now you're all unavailable. There's not a lot of availability out there, right? It's not about energy. Don't want you to get stuck in that idea. You're all sitting there, you're already made. Where are you sitting? In the cytosol, right? I saw somebody asked, that was a good question. Where there was a question, that was one of the answer choices. I like that. Okay. So all of these subunits are sitting in the cytosol. How do we make them available?
Activate them
Activate them, how?
by GTP.
So Mr. Muselli has a good idea, right, by GTP or GDP, but that only works for RAS? For GTPases? What about for alpha and beta tubulin? Okay, Mr. Muselli, he's got I just am going to go until I find the right answer. So that's okay. You would hit on it eventually. So how do you activate something? Did I activate you? I did. I just kicked Nicole. Number 1. What did it do to you? Changed her structure. Didn't it? Want me to kick you again and show you? Did it hurt? I didn't kick her very hard, okay? So when I kicked her, it changed her conformation, and now maybe she's available to bind. And remember, we had this discussion. I'm just repeating it. There's something that activates it. Something is going to change that activates the subunits. Okay? Do we add to existing? Yes, Donna.
For the
Cytochalasin D.
cytochalasin, it only blocks it from adding to the plus end, so it can still add to the minus end,
Yes, but
but it'll just be a lot slower.
it'll be slower and there'll be D caps, right? If we want to, I never ask about G caps or T caps and D caps, but you know what I'm talking about? No? Okay, that's good, because you don't have to know. Are you guys okay over there?
Oh, we're wondering what part is the actin for that?
Right. So let's go back. So, actually, why don't you draw it? You want to come up and draw it? What's your name? It's not really? Jessica. The best way to learn is to teach.
It is.
Oh, it's so hard. You be okay? You don't even need to bring that.
I don't.
No. You can do it from your memory. Jessica, what's your last name? Jessica Chen. This is Jessica Chen, everybody. Let's give her a round of applause. You're not the only one. You know that, right? So what are we talking about?
The Z disc?
The Z-disc. Wow, those are tiny little actins, aren't they? Okay, so, oopsie.
Okay, you said the Z disc was made out of the intermediate
Yes.
filament. So which one's the actin?
Okay, so if I were to draw this, right, look at how small your actins are here. So these are the actins. And let me tell you why I'm drawing it like this, all right? Maybe that will help. So the actins stick out like this. And the myosin is in between. And what is myosin? So the red is the myosin. What is myosin? Does anybody know what myosin is? It's a motor. Okay, we haven't done the motors yet, but it's a motor that moves on actin to the plus end. Wait, no, don't go away. Because now I just realized I made a mistake. You're gonna love this. So, did I answer your question? No.
No.
Jessica, you need to just look at me. I don't bite. Okay, not much. What do you think is the intermediate filament? Just tell me what you think it is.
The green part.
There's two green parts.
The outside. The
Good.
brackets.
It's not the bracket, it's just the straight up and down part. That's the Z disc. That's the intermediate filament.
Okay.
The actins attach to this. Okay? Does that make sense?
You said you made a mistake?
I made a mistake. Okay, so if myosin is a motor and it moves to the plus end, where did I say the plus end was?
Inside?
I said it was over here, right?
Yes.
I was wrong. I hate when that happens, right? Holy crap. How do I do this? Oh, I need the eraser.
Wait, so
Hold on. I'm going to draw it again. Do you want to draw? So put the plus ends on the Z disc. Okay, it's on eraser. Okay, so I originally, this is good, right? It's good for me to find my own mistakes. Poor Richard. Richard, where is your iPad? It's in your lap. He writes notes tiny. I can't even see it. Good. Here are the minus ends. Jessica's got it now. Jessica's totally in with this. Okay, now if I have myosin and it's a plus-ended motor, what does that mean? How the hell does this work? Zilla, you know? No? Okay, you did good. So you got it now? You're never going to forget it, are you?
Wait, does the motor go that way or this way?
Ah, there you go. That's the rub. So here's so Jessica just asked. So Jessica just asked, okay, if this is a motor, does it go this way or does it go that way? It goes both. Okay, so this is as good as I can do. Okay, here's the actin. The myosin sits in between. This is the best I can do. The only thing that can move is the whole sarcomere, and it slides together like an accordion, and it pulls back out like an accordion. So it goes like this. How does that cause, what are we talking about? Okay, we're talking about muscle contraction. Okay, if I draw it like this without all the pretty colors, is everybody going to still understand what's what? All right, so this is what the myosin really looks like. So it has these little heads, and these little heads are all over this thick filament. And we're talking like billions and billions of heads. And then there's a little bit that's not got any heads, and then there's heads on this side. Isn't this pretty? Okay, so the protein itself, myosin, is a protein that has two heads, and it's made of these chains. They're really wound around each other. Okay, it's made up of two heavy chains. So these are two heavy chains, and there's two light chains. And so what structure is this as far as myosin is concerned? Quaternary. Why? Because there's more than one polypeptide chain. How many polypeptide chains are here? 4, 5, 2, 3. Okay, so this heavy is got a long tail, and this heavy has a long tail, and then the two light chains are individual. So it's made up of 4 polypeptides. Okay. Now, these heads bind to actin. Okay? And they are going to move to the plus end. So what does that mean, okay, if I have heads here and I have heads here? Okay, I think of this as doggy paddle. Okay, this is how I perceive this. They go like this. So the only thing that can move is actually this whole thing squeezes in. Okay? So it's an accordion. It squeezes in, and then as soon as we get no binding, what happens? It relaxes. So when I do this, right, I'm ready to punch Courtney in the head. Okay? My muscle is in contraction, right? And then how did that happen? How did that actually get there? I pulled back, but how did that happen? So that's squeezing the accordion together, and now when I punch her, I have to actually cause it to relax. How is all of that happening?
Calcium.
That's all about my brain sending a signal through a motor neuron. What is that motor neuron doing? It's releasing acetylcholine at the nerve terminus. That's causing additional things to happen that we haven't talked about. Okay, and it has to do with calcium in the sarcomeres. Okay? How do you feel about that? Does that make sense? It makes a lot of sense. It totally makes a lot of sense. So you're going to see these are called thick filaments because what you end up with is all of these guys wrap around each other, and you end up with a place in the middle called, you're going to love what it's called, the bare zone. It would only be better if it was the naked zone, right? It's bare because there's no heads there. Yes, Arthur. The sarcomeres get shorter in contraction, and then the myosin remains the same, the actin remains the same. There's no, and you have to be careful here. Remaining the same is not necessarily remaining the same. What does that mean? What the fuck am I talking about? So actin will always be doing what? Treadmilling. So it's changing, but it's not changing. The length is the same, but the subunits are changing. Don't think about that part. Just think about the accordion part. That's probably easier. Yes, Nazila.
So why negative end is inside?
Why is the negative end on the inside? Well,
Isn't it slower?
the negative end is bound to the intermediate filament. It may or may not even cycle. I'm not.
Shouldn't the outside be the problem?
Sorry. See, I really am stuck on that. Minus. Jessica taught me that. See, Jessica? You taught me something today. Look. Yeah. You did? You pointed out, you got me thinking on the right track, sort of. I still am fucking it up, but. Okay. So, why? Nazila, what's the answer to why? Evolutionarily, it works. I mean, you can go to God, okay, right? You can say God made it that way. Or we could go with, how about evolutionarily, this is the way it developed, and it was functional. And when things are functional, well, I try not to fix things that work. When I try to fix things that don't work, then they really don't work, right? Sean?
Yes.
What do I want to, so there's a guy, never mind. Sean,
Okay.
there's another guy. I'm just lumping all the guys with yarmulkes on
Oh.
into one large group. Sorry, that's terrible.
So are the myocyns positively charged?
It has, so, okay, that's really important. I'm glad you said that. So don't say charged,
Okay.
okay? Because that really is not what we're talking about.
Right.
We're talking about moving to an end. So the myocyns can only go towards the plus end. And what does that mean? Is this the only place that myocyns are used?
No.
It's not going to be the only place, okay? There's a lot of myocyns in your body and your cells. So myosin is going to only be able to go to the plus end. Do you remember what I said about actin and its arrangement in the cell? There was a word I used that nobody else uses because it's old. Remember cattywampus? No? I don't think you guys listen to me. I'm just saying. Here's actin. Do you remember me drawing this? Oh, it's coming back. And that organizational scheme, we said, was cattywampus. So now, if you only have one motor, right, this is a little highway. The highway inside of the cell, and I only have one motor, and let's say I need to go from one side of the cell to the other. Let's say this is a minus end here, and this is a plus end, and here's a minus end, and here's a plus end. And I have a motor, and that motor is going down the track towards the plus end on that one. You see what so this guy's moving down here. It could be moving something. Okay? It's carrying something and it's going down the track. Oh, but what happens if I need to change directions? What do? I just jump on another track. Because I can just go right back if I need to. Wait, am I going the wrong direction again?
Yes, it goes towards the plus end, right?
Let's just, it's because I see this as the negative, so I'm going to make that positive. There. It's hard. It's hard to be me. Is it hard to be you? I don't think it's hard to be. Who's getting married this week? Okay, so just don't give me any shit. Look at this list. This is a list of gifts I have to buy. This week, I'm like, how am I going to do that? Holy shit. Along with making an exam and other shit. Ooh, I like this idea. Maybe if there were questions on Moodle, I could use some of those questions so I wouldn't have to spend so much time writing an exam.
Yeah.
See the light bulb come on? Okay, light bulb. All right. So this guy, actin only has one motor. What about microtubules? Let's make a microtubule thing here. We need a different color totally. What color should we pick, Courtney?
Purple.
Of course. Courtney is stuck on purple. Look at her. Can everybody see Courtney? She's dressed all in purple and her hair is purple. Holy shit. Okay. So now, remember the organization of microtubules. Okay, microtubules are way more organized. Where's the plus end? You're like, who the fuck knows now that... Okay, the plus end is on the outside. Okay? What does this mean about motors?
They only go one.
What happens if you have motors for microtubules that only go one way?
They get stuck.
Well, it means everything's going to be either stuck at the plus end or stuck at the minus end. How do you fix that problem?
They can go both ways.
Have motors that go the other way. And that's exactly what happens. Okay, there are motors that go both ways. No, they go one way each. So the motors are called kinesin and dynein. I don't know why, but dynein seems like it just is a negative word to me. That's how I remember it. It goes to the minus end. Kinesins go to the plus end. Is that what it is? I don't know, whatever it is. So kinesins go to the plus end, dyneins go to the minus end. Okay. Guess what the structure of kinesins and dyneins look like? Sometimes there's more. Okay, looks similar, yes? There are heads that are going to bind to the filaments. The tails bind to vesicles. So almost all of vesicular trafficking occurs on microtubules. What else do microtubules do,
I'm
Courtney?
really confused by that picture.
You're really confused by that picture.
So it's got two heads.
Two heads. Just go with two heads and it has some accessory proteins.
Okay.
The kinesin and the dyne.
They're kinesin, so dyneins are a little weird. Dyneins can have three heads. Okay? We're not really covering this in gory detail right at the moment. I'm giving you the bare bones of this. So the idea here is if you need to go towards the plus end, you grab a kinesin and go to the plus end. If you go to the minus end, you need to grab onto a dynein. What do microtubules do in your cell other than vesicular transport? They hold organelles in place. Oh, good. So hopefully now I can get back to the lecture. That was an aside, but it's all part of what we need to know because this is gonna be on the exam. Okay? So I don't think, show this. I'm gonna show this. They're all just little, this is phalloidin, okay? So just in case you are out in nature and you see something that looks like this, don't eat it, okay? I hate to say this, but I have to go there. It has that name
Phalloid. Yeah.
as in phallus, okay? It looks like a phallus. So that's where it got the name. I didn't do any of that, okay? I wouldn't know anyway. Okay. Actually, I just don't want to know, all right? I don't understand it. I don't like it. I don't want it. Okay. The death cap, it has two different toxins. It's not just phalloidin. There's another toxin in this called a manatin. Has anybody ever used that in the lab? This is a pretty common drug that's used in the lab. It inhibits RNA polymerase II. What would that do? What does RNA polymerase II do? Okay, what does a polymerase do? Polymerize. This is an RNA polymerase. When do we make RNA? During transcription. Very, very good. Okay. And what does RNA polymerase II make? mRNAs. mRNAs. What are mRNAs good for?
Translating message.
It's all about proteins. Okay? Making proteins. mRNAs are proteins. Not so, Nazilia, right, it was a good idea. rRNA, it doesn't inhibit rRNA formation because that's RNA polymerase, I think 1. and RNA polymerase 3 is tRNAs. And that could be backwards. God only knows, my brain is not functioning on all cylinders. Okay? So if you treat a cell with RNA polymerase 2 inhibitor, what happens to the cell? You stop making proteins, and eventually you'll die. It doesn't happen right away. But if you want to determine if something, right, if, let's say I infect my cells with Legionella, and I want to see, does it turn on protein synthesis of this protein? Okay, so I treat the cells with Legionella that are alive, right, and I look and see, is there this protein being made? Okay, but I need a control. What if it stops the protein synthesis? So we use this in the lab as a tool, right? What's the mushroom doing? Why does it make all these? Defense. This is self-preservation, people. Okay? Is this going to kill, I don't know, a squirrel that comes along and eats that mushroom? Absolutely. Okay? If you run into a place where you're stuck out in the middle of nowhere and you see all of these big, beautiful mushrooms that nobody's eaten, there's a reason. Okay. All right, let's talk a little bit about microtubule-specific drugs. I alluded to this at the end of the last lecture. I talked about Taxol. So this comes from a tree. It comes from a yew tree. It's the bark, right? And it is good at binding and stabilizing microtubules. So where do we use the microtubules? Cell replication. Okay. So in this case, okay, the other two, nicotazole and colchicine. I don't know where nicotazole comes from. Every semester I look it up, and every semester I forget again. Colchicine comes from a meadow saffron or an autumn crocus. Okay, for those of you who don't know anything, what's a crocus? You don't know what a crocus is? How about saffron? So they're flowers, okay? They're flowers, okay? These bind to tubulin, right, and cause it either to depolymerize. This one causes the subunits to prevent polymerization. Ultimately, these guys, I organize them in the same way. This is going to keep microtubules there, just like what kept actin there. Phalloidin, okay? These two cause depolymerization, just like cytoclasin D and, what was the other one? It was just 5 minutes ago. Latrunculin. Thank you. Is that you? And Angelica? Thank you. Mr. Muselli.
Does that cause depolymerization by preventing it from getting above the critical concentration?
Which one? So colchicine binds the free tubulin, right? So now the free tubulins are no longer free.
So it stays below the critical.
Correct. So now your free subunits is below the critical concentration, so it causes depolymerization. Good. Very good. Okay, here's a cell, and it's undergoing cell division, and I draw this picture for you. Oh, it's pretty. Not very pretty, is it? If those are microtubules, what should be at the ends?
Centrioles.
Centrioles. Okay, and right, I think I already told you that you actually have microtubules going in the other direction. And what we're saying now is Taxol binds and stabilizes these guys. So let's just say Taxol binds and keeps it like this. So now what's bad about that? It can't pull the chromosomes, right? So the chromosomes are attached or not. So if the chromosomes don't get divided, what happens? The cell will die. And so we treat patients. So this is a chemotherapy drug that we give to patients with cancer. And the idea is we're going to kill the quickly dividing cells. But can we just deliver this only to those cells? No. This is the one that we do this treatment and it kills people sometimes. Okay, we give you enough of the drug that we're trying to kill the cancer cells. We don't do it for long periods of time. Okay, but we're hoping to kill the cancer cells and not kill you. Nasty stuff. All right, nasty stuff. So that's Taxol, right? And it came from a tree. It's in the bark of a tree. What's good about that? Protection. Okay, who eats the bark of a tree?
Insects.
Insects? I don't know if it affects insects. That's a really good question. But I don't know about you. We have squirrels that eat the bark of the tree. Okay, there's all sorts of stuff that eats the bark of a tree. Add. Okay. I want you to, so this picture, right, I don't think this is, I don't have any of these linked. I suck, you guys. This was on my list of things to do yesterday. It didn't happen. I unclogged 2 sinks. Let me just tell you, that's not fun. Remember what I said about when things aren't broken, don't try and fix it? So they were broken. They're still a little broken. Now the water can flow down the sink, but they're not all put back together. I never have all the tools that I need at home. So this is an image showing you where the centrosome is. Can you see where that is? On this picture, it's hard to tell. Can you see? What's the word we use for this? What's this? That's the nucleus, so this is called perinuclear. Okay, perinuclear. And what, so if this is the centrosome, what end of the microtubules are here?
Minus?
The minus end. Good. Okay. So if the video was here, what I would show you is these guys, so you treat with the drugs, and then as soon as you wash out the drug, you can begin to see the microtubules form. Well, how would that work? So let's talk a little bit. So microtubules are forming. What are they made out of? Alpha and beta tubulin. And where, okay, if I had shown you the video, you would see that it happens really quickly. I like this. It's like visual imagery in your mind. So what does that mean? These tubulin subunits are sitting around in the cytosol. How can we see it in something like this? Stain it. It's not dark field. Dark field is used for nothing. That I can tell. Right? So dark field is only good at finding cobwebs. You know what I'm talking about? Right? The only way you can see a cobweb is by going like this. Oh yeah, I kind of see it, right? And why are we doing that? Why is your head turning left and right? Because you're trying to see the light refracted off of that little tiny silk web. Okay, when you look straight on, if light is hitting a cobweb, okay, how much is that little silk blocking the light? Not at all. And that's why when you do this, you walk right into the cobweb, because you never saw it. If there's water on the cobweb, or if there's a big black dot on the cobweb, you see the cobweb, mostly. If the big black dot's up here, you are so fucked. Because then you walk into it, and now you have this vague, oh, there was something black up there, which means it's now on your head. Okay? So, right, we turn our heads sideways so that we can see some refracted light. Okay? In this case, what could we do? What could we use to see the alpha and beta tubulin? Not, if it's not stained, tagged. What does that mean? So Ben's got it. So you can attach a GFP to a protein, but you can do it as it's being made. So that means that you take a gene. If I take a gene, so I have DNA. Here's the DNA for beta tubulin. Oh God. Here's the, oopsie. Here's the, when I draw that, what does that mean?
One strand of DNA.
No, Donna, when I draw DNA like that, what does that mean? Don't say one strand of DNA. Really?
Both strands.
That's both strands, okay? Would it ever be, so when we draw, yeah, it's hard, okay? When I draw this for the cell, what did I just draw? The plasma membrane, which is a phospholipid bilayer, Okay, so you have to be careful when you draw this stuff because it all depends on what, right? I never draw it like this. Could I? If I did that, what would that mean? Well, you would have to tell me, I'm drawing the lines and those are the phosphate head groups.
And the lipid tails are in
And the lipid tails are between. Okay, just be careful. If you draw for me a mitochondria. So I've seen people do this. Is that right or wrong?
It depends on how you label it.
It's kind of wrong, people. Okay, because if you're going to draw this membrane
You have to
as a
draw that
phospholipid bilayer, then why wouldn't you draw this membrane as a phospholipid bilayer exactly the same way? You can't have your cake and eat it too. Right? So be careful. What's that, Donna?
That just looks a lot really complicated on
It's complicated. That's why we draw with one line. Okay, so when I draw DNA like this, I'm saying that there's actually double-stranded DNA. Right? And these are A, T, G, C, T, G, blah, 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 blah. Yes? But those are going, right? So this is 5' to 3'. This is 5' to 3'. Okay, what happens if I want to express this protein and I want it to be green all the time? All I have to do is put the GFP sequence upstream, so 5' to this. And I don't have to necessarily make it 5', but it has to be in sequence. So let me just say ATG, blah, 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 blah. How do I make sure that these are going to express a protein that makes a GFP? Maybe I should draw that in a different color. So you're going to have a protein that does this. Okay, here's GFP, and it's going to be linked to beta tubulin. That's what ends up, that's what we're making. Does anybody have any idea how you would do that?
Digest and then insert.
Yeah, you can just insert this in, okay, in the DNA, but it has to be what? These two have to be in frame. Oh, God. What did I say?
In frame.
What does in frame mean, Ben? That's good. It's genetics. So this is like, you're doing genetics. So if things are in frame,
It's gonna have three,
So it's gonna be 3 sets of polypeptides in here.
okay, so each 3 nucleotides is a codon.
Correct. So it's not gonna.
So you can have codon, 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 codon.
You 2 more dots.
Okay, I have 2 more. I have 1, 2, and then if this was part of this, is this in frame or is this out of frame with this sequence? This is out of frame. I need another dot right here. Right? So you read in 3s. So if I put another dot, okay. So I have ATG. What's that code for?
Start.
Don't say start.
Sorry.
It doesn't code for start. It codes for methionine. Okay? Methionine, something, 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 methionine, something, something, something. Okay? If I have this, are you going to say that's start? No, I'll punch you in the head. Okay? Methionine is methionine is methionine. Okay, who's lost? Stephanie's lost. Anybody? Courtney? Okay. Tell me why you're lost, Stephanie. I'm trying to make a protein, and I know that this sequence of nucleotides codes for GFP, which is green fluorescent protein. Okay? We know the sequence of this protein. We know, and we can actually just cut it out of something. We can cut that DNA out and stick it wherever we want. Okay, so if I stick it in front of, okay, if this is the part that's coding for the green fluorescent protein, and I want to express this, but I want to express it as part of this gene sequence, okay? If that, we just need to make sure that they're in, the codons are line up so that we don't end up with, so what we were saying was we were going to have dot dot A, and then there would be TGC, right? And this would be a codon, and then this would be a codon. That would not encode, right? We would lose that methionine. This next one wouldn't be what it was supposed to be. The next one wouldn't be what it was supposed to be. And almost always when you're out of frame, what does it cause? Did Dr. Malone tell you this? It's a frame shift mutation, but it almost always causes a premature stop. Nobody likes premature stops. Okay? Ba-dum-bum. Okay? You got a question, Seth? I answered it?
No, yeah.
Yeah? You don't want to go. You don't want to say anything after that, right? I know. Anybody else have? There was another hand up over here while I was talking. Anybody else? Stephanie, does that make more sense?
It makes much more sense.
Much more sense. Okay, good. Yes.
Are you adding that to an existing sequence?
Yes. And so that's a great question. So that means I also have to know the gene sequence for this protein, and if I said that was beta tubulin, right? I have to know that gene sequence and have it available. We can do all of this now. This is what we do in the lab all day, every day. We take genes that we're interested in, and we express them. And we sometimes link a GFP to it. And why would you link a GFP to a gene?
So you can see it.
So we can see where it goes in the cell. Okay, now, wait 1 second,
I have
Melissa.
a question.
Hold on.
Okay.
Let me just ask you a question. If I express this GFP, and it's just the same size, and it's big, it's 23 kilodaltons, and it's the same size as this, what might you guess would happen to the protein of interest that we're interested in?
It'll be double the size.
It's going to be double the size, but what else might happen to it?
Function map.
The function could be totally fucked up. Right? I'm saying you're making this big protein and you're linking it to this protein. How do you know that when you express this, it's not going to, right, bind like that to the protein of interest? How do we know? We don't. We have to test it. We have to make it and say, does it do the same thing that the normal protein does? And if it doesn't, then we can't use this technique. Okay? Somebody else. Was it you, Courtney? Yes. Go ahead.
So my question is, why are there 3 dots and then the A?
Okay. 3, 3, 3, 3, 3.
Okay.
And then ATG. Now is a codon. CTC. This was the beginning of my beta tubulin.
Okay. I just
The boxes are off now, right?
Okay.
So that was without the dot.
Oh, okay.
Does that make sense?
A little bit.
A little bit, not a lot of it.
Yeah.
Yeah, so the box.
Okay, I'll take that dot back out, Courtney. Okay, so there's only 2 dots. Okay, so now there's 2 dots. 1, 2, 3. This would be the codon if we didn't have that extra dot in there.
Okay, and that's when
That's a
it's
frame shift.
frame shift. Okay, I see.
Okay, I think we've done enough for one day, enough damage for one day. Yeah, I would go read those questions on Moodle. That's what I would do. Just say, yes. So the question, hold on, the question is, can we download the exam yet? Has Mary Pat made the exam? No. I haven't made the exam. I haven't even begun thinking about making the exam. Maybe tonight.
Just
I'll email you all, okay?